Our biocuration dialog system is available at dialog.bio. Here I will use the SVG-MViz interface, which is optimized for interactive modeling coupled to dialog. I will show an example of learning about mechanisms by which pancreatic cancer cells proliferate and develop resistance to drug treatment, and through dialog I will learn about possible resistance mechanisms and build a model of my hypothesis. I described my initial modeling goal, which is to find out how KRAS increases cell proliferation in pancreatic cancer cells. I then ask about a potential mechanism, does KRAS activate MEK, and find that there is literature evidence for this mechanism, and I can in fact drill down and look at what that evidence is. Much of the knowledge of the machine comes from a system we developed called Indra. Indra integrates literature-scale biomedical text mining and expert-curated pathway databases through a process of knowledge assembly. We've processed over 25 million abstracts and 3 million full-text papers with 6 machine reading systems, and continue to run these systems in the cloud on around 4,000 new publications that appear every single day. I can now start adding mechanisms to the model using natural language, and at each step the machine interprets this and incrementally assembles an executable mathematical representation of these mechanisms. It can also give suggestions on mechanisms to add and point out possible causal gaps in my thinking that I can then refine as needed. The machine also detected that I reached my initial modeling goal and gave me a summary of the path connecting KRAS with cell proliferation. I can now find a drug that targets MEK, add it to the model, and ask about its effect on cell proliferation. To answer this, the system runs a model simulation corresponding to my query to show that, as expected, this drug fully inhibits proliferation. However, this is not consistent with empirical observations due to a feedback loop which upregulates a parallel pathway to proliferation through SHP2. Jumping ahead, I have now extended the model and can now ask if trametinib still fully inhibits proliferation despite the feedback. As we see, proliferation is no longer fully inhibited. Let's now find a drug that specifically targets the feedback mechanism and confirm that it is sufficient to stop proliferation. I first ask for a drug that targets SHP2 and find that there is this one drug that I can then add to the model. Now I can check whether proliferation is always low, and this again is answered by the system using model simulation. In the end, we confirmed that combining trametinib with SHP2 inhibitor is sufficient to inhibit proliferation. As we saw from this example, the machine agent provides a vast biological knowledge base which is automatically extracted and assembled from the scientific literature, and combines this with the ability to model novel hypotheses and their implications. Altogether, these capabilities enable scientists to formulate new ideas and make discoveries by communicating with a computer.